it's a real honor to give the NSIG lecture. John, as Alan said, was really a fantastic physician scientist and clinical researcher. And as you'll see, a lot of my career has been based on studies in a clinical research setting, really trying to understand physiology using genetics and pharmacologic probes to really try to understand. So let's see. So this is my title, and I do have some disclosures. I consult for several companies developing medicines in the kind of space that I'm going to be talking about, and specifically, I'm an inventor on a patent related to lamidopide, a drug that I'm briefly going to talk about that's owned by Penn and licensed to Alnylam Pharmaceuticals. So there's a lot of talk about translational medicine and translational therapeutics, and at Penn, several years ago, we tried to really define what we mean by that, and of course, there's a lot of great basic research that occurs here in Penn and everywhere. You really need to take that basic research and prove that these concepts in cells actually are relevant to relevant animal models. And then ultimately, the very hard job of proving that these pathways and concepts are relevant in humans. Of course, this is the kind of clinical research center type really generates new insights that really come back into this paradigm, allow us to really test pathways that we didn't previously suspect were relevant to the disease we're interested in, and then move things down this paradigm. And what I'm going to do with this lecture, I'm not going to dive into any specific story in great detail, but really try to give you multiple examples from my field, lipoprotein metabolism and atherosclerosis, how this pathway of the study of human genetics has actually literally generated insights into new therapeutic targets which are moving their way along this path toward ultimately being approved to treat people with unmet medical needs. So this concept of employing human genetics to really lead to smarter and faster development of new medicines is one that the field has widely embraced. A lot of academic interest in this and tremendous biopharmaceutical interest in this concept that human genetics really can be leveraged to identify new pathways, to validate targets that we're already interested in, and to ultimately give us greater confidence that, in fact, if we target a certain pathway, we're likely to actually have the kind of benefit that we hope we have when we move that new medicine into humans. So I kind of like this slide. I just thought I'm going to be focusing on heart disease and atherosclerosis. And this is a slide that I found from WHO that basically lists the major cause of death in each country around the world. And you can see that heart disease is the major cause of death, not just in the U.S., which we talk about a lot, but in many countries around the world. It is still, despite advances that have been made, the major cause of death worldwide. So my focus for this talk is going to be how gene discovery is leading to insights into new biology. I'll give you a few brief examples of that. And also insights that lead to new therapeutic targets. And that's really the general organization of what I'm going to be talking about today. And I'm mostly going to talk about so-called ApoB-containing atherogenic lipoproteins, the lipoproteins that are bad, that actually do directly promote the development of the atherosclerotic plaque. Of course, LDL I'm going to spend some time on, very familiar to you, but there have been a lot of new developments. Then I'm going to turn to triglyceride-rich lipoproteins, the lipoproteins that have more triglyceride, for which there have been a lot of questions, but I think the data are now absolutely incontrovertible that they are also causally related to the disease. And I'm not even really going to talk about Lp(a), another area where the genetics has played a huge role in inferring causality of this particle for the development of coronary disease. If we have time, I'm going to briefly touch on HDL, a topic of great interest to mine, but my major focus is going to be on LDL and triglyceride-rich lipoproteins. And so LDL, the bad cholesterol, you're all very familiar with it. Like a lot of quantitative traits, LDL is partly genetically determined, and this is the best estimate based on heritability. About 50% of the variability in LDL levels in the general population is genetically determined. So that really leads the question, what are the genes that actually impact on LDL level in the general population? So if this is the distribution of LDL levels in the general population, we can take one of several approaches. And of course, all these approaches have been taken, and I'll give you some examples. One is to study people at the extremes, people with extremely low levels of LDL or extremely high levels of LDL, people who are very likely to have monogenic or Mendelian causes of low or high LDL. But we can also do genome-wide studies where we look at LDL levels in the general population and ask which variants at which genetic loci affect LDL. And I'm going to give you examples of both of those and how they've informed the biology and the new therapeutic targets. So the concept for the genome-wide approach, and I'll come back to some Mendelian issues in a few minutes, is that we look at genetic variants across the entire genome, and then we look, in this case, for which of those variants are highly significantly, genome-wide significantly associated with LDL cholesterol levels as a trait. So this is really looking for a needle in the haystack. We're looking for these relatively few variants throughout the genome that are very, very highly related to LDL. A little like this cartoon that I like, the scientist looking at his jigsaw puzzle of 3 billion pieces and his colleague saying, I think I found a corner piece. That's a little what this exercise is like, sifting through the entire genome, looking for those corner pieces that really are of particular interest. And so I'm going to start with an example that I think is instructive, just for sake of trying to illustrate what I'm really trying to communicate today. And that is that this is a genome-wide association study that we participated in, led by my colleague and friend Seymour at the Broad Institute. And this was published now several years ago, but was 95 discrete loci throughout the human genome associated with one of the 3 major lipid traits, LDL, HDL, or triglyceride. And I've circled HMG coreductase, I think familiar to many of you. So common variants at the HMG coreductase gene are associated with LDL cholesterol. Now, these common variants are associated with very modest differences in LDL, 2 or 3 milligram per deciliter. But because of the huge size of the study, over 100,000 people, that finding is statistically significant. So I'll come back to that. Many people have argued that these studies are worthless because these changes are very small. These differences are very small. And I'm going to argue that, in fact, the size of the effect for these studies really doesn't matter. So HMG coreductase, it turns out, not only are these variants associated with LDL, but they're independently and directly associated with coronary disease. And that's a second concept that I really want to drive across with regard to the human genetics, is that we can begin to infer causality from human genetics. Since these variants, which affect LDL, also affect or influence coronary disease, we can begin to infer that perhaps LDL is directly causal for coronary disease. Well, of course we know that. We know that because of all the intervention studies with LDL. But the point I really want to make is that, of course, HMG coreductase is a known gene. So the fact that this was picked up in genome-wide association is perhaps not so interesting. But biochemical studies from decades ago identified this as the rate-limiting step in cholesterol synthesis, as I think many of you know. And so as a result of that biochemical identification, of course, statins were developed. There are now 7 statins on the market. They all have this general structure, which is very similar to HMG coreductase. They directly inhibit the enzyme, and they block cholesterol synthesis. And we all know that statins very effectively lower LDL. They very effectively reduce risk of coronary disease. So the greater the reduction in LDL, the greater the reduction in coronary disease risk. And so this is an example where the drug preceded the genetics. So based on the biochemistry, the drug class was developed, now the most widely prescribed class of drugs in the world. And so I like this kind of way of thinking about things. This is an example where the pharmacology got developed. Statins clearly lower LDL. They clearly reduce risk of coronary disease. Then the genetics came along a couple decades later. Common variants at HMG coreductase reduce LDL and reduce coronary disease. And I think, again, my point about effect size is these common variants are very minor in terms of their ability to reduce LDL. And frankly, very minor but statistically significant in reducing coronary disease. But a drug that targeted this pathway much more potently, much more substantially lowered LDL, and of course very substantially lowered coronary risk. So I want you to imagine that if this biochemistry hadn't been discovered, and this was the first identification of HMG coreductase based on the genetics, it would have made it a very attractive target. And that's really the concept I'm going to really proceed with is we're in an era now where we're discovering a lot of additional genes that have this kind of relationship, and they become very interesting, attractive targets for the development of medicines that target those protein products as a way to try to treat disease. So I'm going to move to a second example that is also an existing pharmacologic tool, medicine, just for sake of illustrating this point once more, and that's NPC 101. And I think many of you probably know NPC 101 is a transporter in the gut. It promotes cholesterol absorption via the gut into chylomicrons and ultimately gets it back to the liver. And as I think many of you know, the drug ezetimibe inhibits NPC101 and actually reduces cholesterol. So what's interesting is the genetic variants of NPC101 that associate with LDL, again, recently have been shown to associate with coronary disease. So loss of function variants in NPC101 that reduce LDL levels, reduce risk of coronary disease. And of course, the drug ezetimibe, which inhibits NPC101, has recently also been shown not only to lower LDL, but to reduce risk of coronary disease. So I give you these two examples briefly to point out that the genetics and the pharmacology line up very nicely. In this case, loss of function mutations in NPC101, lower LDL and lower coronary disease, and of course we now know that the drug does the same. So these are two known drugs that target LDL that are proven to reduce coronary disease. And I want to point out that the major drugs that we have at our disposal for lowering LDL really converge on the LDL receptor pathway. That is statins, I've mentioned, cholesterol absorption inhibitor, ezetimibe, a third-class bile acid sequestrants, all converge ultimately on upregulation of the LDL receptor in the liver as the mechanism by which LDL is being reduced. So very effective. We really thought we knew an awful lot about the regulation of the LDL receptor, but as I'll tell you in a few minutes, the human genetics pointed to a whole new mechanism for regulation of the LDL receptor that is leading to a new therapeutic. But first, I want to make the case that, in fact, there's still major unmet medical need in the treatment of elevated LDL, despite the availability of statins and ezetimibe and bile acid sequestrants. This is kind of a conceptual pyramid. At the top are patients with homozygous familial hypercholesterolemia, the tip of the iceberg. I'm going to come back to that disease in just a minute. But there's a whole other group of individuals who have high LDL despite maximally tolerated therapy. Of course, a large group of people who are statin intolerant, can't take statins because of muscle pain. And these individuals, particularly those who are higher risk or on maximally tolerated statin, need additional therapies. We don't have adequate therapies currently for these individuals. In fact, to illustrate that point, something we use for the most severe patients is something called LDL apheresis, a procedure that, frankly, is a little barbarian, but it's really all we have for a lot of these really severe patients, where they come in usually every 2 weeks, they get their red blood cells separated and removed back to them, they have 2 lines put in, then their plasma is passed over these columns which specifically bind up the LDL, and then the LDL depleted plasma gets returned back to the patient. We have the biggest program in the country for this, about 32 patients currently getting LDL apheresis. But I think this illustrates that there's unmet need. If people are coming in to have this done every 2 weeks for about $3,000 to $4,000 a procedure, it's clear that there's unmet medical need. I just thought I'd show you this is what it looks like. So this is the patient hooked up to the instrument. This is what the column looks like before the procedure and what the column looks like 3 hours later. This is all the LDL in their blood being bound up by these columns. So clearly there's unmet need. There's been a lot of interest in developing new therapies. I'm going to tell you interesting vignettes about 2 new therapies that are now recently approved that were based on the study of people with rare inherited very low levels of LDL. So this is an example of a Mendelian syndromes of very low LDL leading to new insights that ultimately led to new therapeutic targets. So the first is a disorder that I spent a long time studying when I was at the NIH, A-beta lipoproteinemia. These are very rare patients, an autosomal recessive disorder, which at the time in the early 90s, what we knew by studying these patients in the Clinical Research Center are that they had no levels of, no ApoB, no LDL in their blood, and no ability to actually secrete VLDL, the precursor to LDL, from the liver. So they had a block in secretion. We didn't know why. A large consortium led by Dick Gregg ultimately found the molecular mutation, and it's a gene called MTP, microsomal triglyceride transfer protein. It's an ER protein that loads the triglycerides onto the ApoB protein, permitting the assembly of the VLDL particle and ultimately its secretion. So this was a really interesting biological finding, put a new protein on the map that's critical for VLDL assembly, but also led to the idea maybe inhibiting MTP would be an interesting strategy for reducing VLDL secretion and reducing levels of LDL. In fact, Dick and his colleague John Wetterau went to Bristol Myers Squibb, developed the world's first program for developing small molecule inhibitors of MTP, and were successful in developing a drug. I had moved to Penn by this time, and I worked with them to study this in the Clinical Research Center, and actually proved in a small, relatively short proof of concept study that this drug, now called lamidipide, markedly reduced LDL levels in patients with hypercholesterolemia compared to placebo over 4 weeks, as you can see here. So this was exciting. It was proof of concept that inhibition of MTP pharmacologically reduced LDL. There were some issues with this study, which I'll come back to, which led Bristol Myers Squibb to basically abandon development of this drug. They basically said, we don't think this is going to compete with statins. That was their orientation at the time. And we're not going to develop it any further. And I said, I really think that there's certain patients who could benefit from this drug. Will you allow me to continue to develop it for those patients? And those patients are patients with homozygous familial hypercholesterolemia. This is a 12-year-old patient of mine from several years ago. She had developed as a child these xanthomas on her knees and elbows. If you biopsy these, they'd be all macrophages loaded with cholesterol. Her cholesterol was, LDL cholesterol was about 760. And at playing soccer one day, she developed chest pain. We took her to the cath lab at Children's Hospital. She had a significant left main stenosis and had bypass surgery at Children's Hospital. So this is classic homozygous FH. She has no LDL receptor, and she basically has markedly elevated LDL, and there really is not good therapy for this. So I thought maybe this drug would be good for this disease. Again, these patients have lack of LDL receptor, and therefore they don't really respond to statins and bile acid sequestrants and ezetimibe because, of course, they work through upregulation of the LDL receptor. So we needed a different approach, and I thought lamidipide might be a good way to go, because by inhibiting MTP, it would reduce the secretion or input of LDL and bypass the fact that these patients had no LDL receptor. So to their credit, Bristol Myers Squibb actually transferred the asset to Penn, allowed us to continue to develop it. And long story short, we actually carried out a phase 3 registration trial in 29 patients, basically showing about a 50% reduction in LDL that was persistent. So this actually permitted registration of the drug, which was approved about a year and a half ago now. So it's on the market for this very specific indication, of orphan indication, of homozygous familial hypercholesterolemia. So that's one. A second related story, it relates to another Mendelian cause of extremely low LDL, and that is familial hypo-beta-lipoproteinemia. So this is a disorder in which truncating mutations in ApoB result in shorter ApoB proteins that don't assemble the VLDL correctly. And so in a similar fashion, these patients don't have adequate VLDL production and have low levels of LDL as a result. So this led, and I'm going to keep this story very short, but this led to the idea that maybe targeting ApoB therapeutically would be interesting. Now, this is not a small molecule target. But ISIS Therapeutics, the leading company for antisense oligonucleotide therapeutics, to their credit, several years ago decided to target ApoB using an antisense oligonucleotide, which is now called mipamersen. And they carried out a very similar development plan where they focused specifically on homozygous FH. And their phase 3 trial showed, compared to placebo, about a 25% reduction in LDL with mipamersen, targeting ApoB. So this drug was approved about a month after lamidipide was approved. And now both of these are on the market for this very orphan indication of homozygous familial hypercholesterolemia. So I think it's very interesting. Based on the human genetics, these drugs both were developed, both target different pathways of VLDL production, and both lower LDL and are approved for patients with homozygous FH. So it's sort of an interesting way that we can use the Mendelian disorders to infer new biological pathways that are potentially new targets. Now, these drugs have one major issue that really precludes them from being used more broadly, and that is that, as you'd expect, a mechanism-based increase in triglycerides in the liver as a result of inhibiting VLDL secretion. So VLDL has triglycerides, of course. If you're not secreting VLDL as effectively, those triglycerides get left behind and accumulate. So there is an increase in hepatic fat. We don't know what that means. We'll have to see over the long haul whether that's an issue for the patients, but it's a risk-benefit. So the decision was made by the FDA that in this very severe homozygous FH disease, where they have very few other options, that it's reasonable to consider treatment with these drugs. So we'll see. So I'm kind of filling out this chart. You can see for these, the genetics certainly tell us that these genes are related to LDL. We don't really know about coronary disease. These conditions are too rare. And now we know the pharmacology lowers LDL. Of course, we don't have data related to the pharmacologic approach and the impact on coronary disease. So these two drugs are now targeted toward homozygous FH, but that still leaves us with this huge group of individuals who don't have adequate therapies, and there's still unmet medical need. And that really takes me to the next story, which this story is often held up as one of the poster children of the idea of human genetics identifying new biology and new targets. And it's the story of PCSK9. Probably familiar to some of you, but I suspect not all of you. So this story started when investigators in France had patients with really high levels of LDL, but didn't have mutations in the Ldl receptor, or frankly, any of the other two genes that are known to cause high LDL. So they did classic linkage analysis, and they eventually honed down to a gene called PCSK9, which virtually nothing was known about at the time. And they found mutations in that gene that were clearly associated with the high LDL in the family. And we now know, we didn't at the time, but we now know these are gain-of-function mutations in this particular protein. But again, this put PCSK9 on the map as something related to LDL, something that we really previously had no idea was the case. And then Helen Hobbs and Jonathan Cohen did a brilliant experiment. They have this Dallas Heart study, and they reasoned maybe mutations in this gene cause low LDL. So they took individuals at the lowest levels of LDL in their bell-shaped curve of LDLs in the Dallas Heart study, and they sequenced PCSK9 in these individuals and got kind of lucky, but it was really a terrific experiment. In the Caucasians in this study, 3% of them had LDLs, had a mutation, R46L, that lowered LDL by 20%. And then in the African Americans in the study, they found two different mutations, nonsense mutations, these two, each of which lowers LDL by about 40%. So amazingly, they found that mutations that clearly reduced function also reduced levels of LDL. And then what really sealed this is they went to a much larger study, genotyped or looked for these mutations, particularly in the African Americans in this larger study, and found that they were markedly associated with reduced risk of coronary disease. So here are loss-of-function mutations in a gene we know almost nothing about that reduce LDL and reduce risk of coronary disease quite dramatically. So even though we knew almost nothing about the biology at the time, it immediately put this on the map as a potential target for new therapeutic intervention. A beautiful reciprocal story of gain-of-function mutations raising LDL, loss-of-function mutations lowering LDL, and of course protecting against coronary disease. So this put this protein on the map. We really thought we understood LDL receptor regulation pretty well. Of course, Brown and Goldstein won the Nobel Prize for discovery of the LDL receptor. It's been intensively studied. And what we thought we knew is that the LDL receptor shown here binds LDL, targets the endosome where it dissociates. The LDL goes to the lysosome and is degraded. And then the LDL receptor recycles back to the cell surface where it can continue to do this thing multiple times. And a lot of knowledge about the transcriptional regulation of the LDL receptor. But it turns out that PCSK9 regulates the LDL receptor post-translationally. PCSK9 is a secreted protein, secreted by the liver. It binds to the LDL receptor, and it's internalized along with the LDL. And instead of allowing the LDL receptor to dissociate and cycle back, it targets the LDL receptor also to the lysosome. So think of it as basically scrubbing the cell surface of the LDL receptor and controlling LDL receptor at that post-translational level. So this led to the idea, if you could inhibit PCSK9, you could potentially increase LDL receptor density and lower levels of LDL. And sure enough, monoclonal antibodies have been developed that bind the PCSK9, prevent it from interacting with the LDL receptor, and result in substantially increased LDL receptor in the cell surface. And this was the first report in the medical literature of a PCSK9 antibody in humans. And you can see that there's very substantial reduction in LDL, up to 60%. After a single dose of a monoclonal antibody targeting PCSK9. So as I'm sure many of you know, this is an incredibly hot target. There's been a lot of interest and discussion about this. There are two of these are leading the way, a Sanofi Regeneron antibody and an Amgen antibody. And some of you might know that both of these are in late-stage phase 3. In fact, they both have their FDA advisory committee meeting in June, next month. And there's expectation that they're likely to be approved by third quarter of this year, so in just a few months. So tremendous excitement about this class, again, discovered because of the human genetics. I thought I would just make one other point about biological approaches. Alnylam Pharmaceuticals is the leading siRNA company that is focused on using RNA interference for therapeutic purposes. And again, I don't have time to go into the details, but they package their siRNAs in delivery vehicles, and then these siRNAs target specifically mRNAs within the cell type, in this case the liver, and cleave it and actually prevent that mRNA from being translated into protein. So they developed an siRNA therapeutic against PCSK9. They've presented and published their initial phase 1 work in humans. And the bottom line is it lowers PCSK9 levels in the blood, shown here, and it lowers LDL levels, shown here. So this is yet another biological approach to trying to reduce PCSK9. So tremendous enthusiasm for this field. Of course, the genetics showed that PCSK9 loss-of-function mutations lower LDL and protect against coronary disease. The pharmacology shows that they lower LDL. Of course, we don't know this yet. This is the big question. But based on the human genetics, there's very high confidence that, in fact, these drugs will lower risk of heart disease. And in fact, the FDA, I think, is very likely to approve these drugs, even though they don't yet have definitive phase 3 data that heart attack and coronary disease is reduced, in large part based on the human genetics. So again, a poster child for this whole approach. So I think if we look toward the relatively near future, we have PCSK9 inhibitors really allowing us to much better treat this group of individuals. And you could argue we're starting to get to the point where perhaps we've got the LDL field covered. And so that may be the case, but I just want to conclude this section on LDL by pointing out that there are a lot of the loci for LDL that have been discovered by Genome-Wide association that are basically unknowns. They're loci where we really had no idea that they were in any way associated with LDL metabolism. The ones that are shaded are kind of known genes that we had prior knowledge of, but the ones that are unshaded basically are sort of novel biology. So one of my big interests scientifically is looking at these lists and trying to identify which ones do I want to work on to try to better understand the biology that explains why that particular gene is associated with LDL cholesterol. And I'm just going to give you one very brief vignette that relates to one locus that we've worked on hard now for several years. And so what we did several years ago when we got that list is we ranked the loci by p-value as a sort of a surrogate for how strong the association was with coronary disease. And these were the top 6. You can see some nice friends here like Ldl receptor and ApoB and HMG corereductase. Nice to see, positive control. And of course, we knew about ApoE and ABC G5 and G8 as well. But the top one on the list, a p-value of 10 to the minus 160th, was a locus on chromosome 1 that did not harbor any genes that were known to affect LDL. And what was interesting about this locus is the common variant was a minor allele frequency of 25%. The homozygotes for that variant had lower LDL. And what really got us excited is independently, GWAS for coronary disease had identified this locus as highly significantly associated with reduced risk of coronary disease. So this looked to be pretty important for human biology. Here's a locus where variants reduce LDL and reduce risk of coronary disease coronary disease. So like a lot of these studies, I want to give you a flavor for sort of how one goes from here. This is a complex locus. The top SNP is right here. There are at least 4 genes that are kind of in this ballpark here that are potential candidates. It turns out that carriers of the minor allele, the homozygotes, have higher levels of expression of 3 of these genes at the locus. And those are the people, of course, who have the reduced levels of LDL. So using this, we kind of reasoned maybe if we got the right gene and we expressed it in mice, we should be able to reduce LDL levels in mice. That was the idea. So Alana Strong, an MD PhD student in the lab, did that experiment with several of these genes, and she found that a gene called sortilin actually markedly reduced LDL levels. This is the LDL peak here when expressed in mice. And that really gave us a clue that this was the causal gene at the locus. And I'm not going to go into great details, but I just want to summarize an awful lot of work by pointing out that overexpression of sortilin reduces LDL in mice, knocking down sortilin with siRNA increases LDL in mice, and this gave us a lot of confidence that this was the causal gene at the locus. And really, again, sortilin had been studied in the central nervous system, but never studied in the liver. There wasn't a single report of sortilin in the liver. This is a transmembrane protein. It's a member of a larger family that has these so-called VPS10 domains that bind multiple ligands. And so this really led us to ask the question, how does sortilin in the liver actually affect LDL metabolism? And of course, we used what was known about its biology. It's a trafficking receptor that can bind ligands in the Golgi and traffic to the lysosome. It can also go to the cell surface where it can bind ligands extracellularly and traffic them to the lysosome. So I'm going to summarize an awful lot of work in a very short slide here by saying that sortilin has complex roles in LDL metabolism. It can bind VLDL, newly synthesized VLDL in the Golgi, and rather than allowing it to be secreted, can target it to the lysosome for degradation, so limiting VLDL secretion. And at the same time, we showed that it can actually bind LDL. It's another LDL receptor. It can bind LDL and target LDL to lysosomal degradation. So the combination of those two things, effects on secretion of VLDL and effects on catabolism of LDL, really led to the effect. And remember, the genetic variant is gain of function. These people have much higher expression of sortilin in the liver, so they have even further reduced VLDL secretion and further uptake of LDL, leading to the low LDLs in the blood. So this led a whole new biological pathway. Is it therapeutically tractable? Maybe not. We would have to increase sortilin in the liver to really employ this therapeutically. Not out of the question, but obviously a difficult thing to do from a therapeutic standpoint. But we're still very interested in what regulates sortilin, and maybe there is an interesting therapeutic approach to upregulating sortilin. So I'm going to leave LDL, and I'm really going to turn to what is really one of the most interesting areas now, and that is triglyceride-rich lipoproteins. So these are not LDL now. These are VLDL and VLDL remnants and IDL and chylomicron remnants, the particles that come from both the intestine and the liver that have triglycerides in them. And of course, we pick up on this clinically by measuring triglycerides, usually fasting triglycerides in the blood. There's been a long history of questions about whether triglyceride-rich lipoproteins are causally associated with coronary disease. And I just say that the human genetics, I think, have absolutely established that the answer is yes, that triglyceride-rich lipoproteins are causally related to the development of coronary disease. I'm going to just briefly, in the interest of time, mention that, again, for new biology, one of the genes we've looked at in substantial detail is an interesting gene called TRIB1, which is associated with all three of the major lipid traits, including triglycerides. And you can just get a sense here for the variants at this TRIB1 locus, very strongly associated with triglyceride levels. And I'll just summarize by saying that these TRIB1 variants have strong associations with triglycerides and other lipids. Independently, they've now been shown to be strongly associated with coronary disease. And interestingly, they're also associated with liver fat and liver enzyme elevations. So, in fact, the variants at this locus have real importance to lipid and liver lipid metabolism and actual coronary disease. So this is another one where we're very interested in the biology. I think in the interest of time, I'm going to skip a few slides, except to say that the knockout mouse has markedly increased liver fat, steatosis, consistent with this idea that TRIB1 is affecting liver fat metabolism. And to summarize, the mice overexpressing TRIB1 have a reduced lipogenesis, reduced formation of new lipids, new fatty acids. The knockout mice have increased lipogenesis. So there's a clear role of TRIB1 in lipogenesis, and we have really been able to focus this on its effect in regulating the transcription factor CBP alpha. So TRIB1 is a degrader of CBP alpha. It targets CBP alpha for degradation, and CBP alpha in the liver we found is a major regulator of lipogenic gene expression. So again, just really briefly summarizing how the genetics led us to this new biological pathway. And here we are very interested in the concept that pharmacologic upregulation of TRIB1 in liver might reduce hepatic steatosis, reduce atherogenic lipoproteins, and reduce risk of coronary disease. And this is one that we are actively working on to see if we can develop a therapeutic approach to upregulating TRIB1 in the liver. So we'll see. But where I want to go with triglycerides now, and kind of the remaining time we have, is really to emphasize that multiple different loci, variants at those loci that have been shown to affect triglycerides, have also been shown clearly to affect coronary disease. Now, this is very strong genetic evidence that, in fact, triglyceride-rich lipoproteins are directly causal for coronary disease. And I want to make one comment about perhaps the classic triglyceride metabolizing enzyme, lipoprotein lipase. So common variants at lipoprotein lipase clearly came up as associated with triglyceride levels. Okay, that was interesting. I'm sorry. Oh my gosh, I'm so sorry. I just crashed PowerPoint here. All right, give me a second. So I'll just say what I was about to show you. So lipoprotein lipase hydrolyzes triglycerides and triglyceride-rich lipoproteins. Let's see here. Yeah, I'm still a Phillies fan despite their miserable season. How are the Mariners doing? I haven't been keeping track. I'm sorry. Okay, here we go. Okay, I'm not sure what happened there, but all right. Okay, so lipoprotein lipase is an enzyme that hydrolyzes triglyceride-rich lipoproteins. And very interestingly, there's a gain-of-function variant in lipoprotein lipase that is 10% allele frequency in the general population. It lowers triglycerides, and it actually protects from coronary disease. So I think this is a great example of how a common variant that increases lipoprotein lipase activity is protective. And what's really been interesting over the last couple of years is there are multiple genes that are on this list that influence lipoprotein lipase activity and influence risk of coronary disease. And I'm going to tell you one vignette that I find really exciting, and that's related to ApoC3. So we've known about ApoC3 for a long time, but it's really coming back in terms of the interest in the biology. We've known that ApoC3 has the ability to inhibit lipoprotein lipase. It's an endogenous inhibitor of lipoprotein lipase. And really exciting, last year in New England Journal, 2 back-to-back papers from Say Kathriesen's group in Boston and Ann Tyberg-Hansen's group in Copenhagen, but came up with very similar results. That is, by sequencing ApoC3, or in this case exome sequencing finding ApoC3, they found mutations that lower triglycerides and reduce risk of cardiovascular disease. So very similar to the PCSK9 story. Loss of function variants that lower, in this case, triglycerides, not LDL, and actually reduce risk of heart disease. And so there's been a tremendous interest in trying to understand how ApoC3 actually affects triglycerides, and then the possibility that these may be a clinical target. So So one of the interesting things about these studies is that it's always helpful to identify people who completely lack the gene. I didn't tell you, but for PCSK9, everything I talked about was heterozygotes, but in fact, there have been now several homozygotes identified for PCSK9 mutations where they literally lack PCSK9 completely. And you know what? They look like they're pretty much fine. They have very low levels of LDL, but no other issues. And that was very reassuring to the pharmaceutical world, because they really felt that you could really suppress PCSK9 a lot and not have a problem. But so there's been a lot of interest in the same thing for ApoC3. These heterozygotes exist, but what about homozygous, essentially knockout humans? And despite sequencing and genotyping well over 100,000 people of European descent, there really haven't been any homozygous knockout patients identified. So I have a colleague, Danish Salehin, who is a remarkable guy, an MD from Pakistan, did his PhD in the University of Cambridge, and has recruited about 60,000 individuals out of Pakistan for studies related to cardiovascular and metabolic disease. And what's interesting is there's about a 40% rate of first-cousin marriages in Pakistan. So you can imagine that the probability of finding homozygotes in a group, in an ethnicity like this is much higher. And in fact, out of 7,000 people we sequenced, we found 25 homozygotes. And I'm just going to tell you a vignette that relates to a clinical research center type study. So we identified one of these individuals in a small fishing village about 3 hours north of Karachi, and Danish sent his team out there to recruit his big extended family, about 27 individuals from the family, a lot of intermarriage, as you might expect. So this is the nuclear family. This is the proband. His triglycerides are 62, low. He married his first cousin, as shown here, and they have 9 children. And what we found is, in fact, unexpectedly, his wife, his first cousin, is also a complete deficient patient. Triglyceride is 67. And of course, all 9 children are completely deficient in ApoC3. So I give you this as an example of how we went from like literally no patients in the world identified to this one nuclear family of 11 individuals And in the entire extended family, of course, the homozygotes have undetectable C3 and very low levels of triglycerides. But the one remaining thing we did is we actually gave them an oral fat tolerance test, a high-fat milkshake, to look at the effects. And this is basically a kind of study where you give the milkshake, you draw blood at several time points. The normal response is to basically have the triglycerides go up over time when you give this high-fat milkshake. This is the wild-type people with no mutation. And these are the individuals with no C3. They have virtually no increase in their triglycerides after being given this very high-fat milkshake. So we think this probably has something to do with the protection from coronary disease. There's a lot of interest in the lipoproteins that occur after a high-fat diet as being particularly pro-atherogenic. So this is a very interesting target for new therapies. ApoC3 has been targeted by antisense oligonucleotides, by, again, ISIS. And they are furthest ahead in this, and they've actually published some of their data. This is their antisense oligonucleotide at different doses, reducing ApoC3 levels in the blood and reducing triglycerides in the blood. So sort of proves the concept that if you target this pharmacologically, you can reduce triglycerides. So we're excited about this. We think this is orthogonal to LDL reduction. There's still a lot of people with increased cardiovascular risk who have high triglycerides and normal good LDLs. And so this might be an orthogonal therapy that could further reduce cardiovascular risk. But really, to take this even further, there are additional genes coming on the scene. ANGPTL3 and ANGPTL4 both inhibit lipoprotein lipase, and ApoA5 is a stimulator of lipoprotein lipase. And there's genetic data, very strong genetic data for all of these in terms of affecting triglyceride levels and increasingly for their direct effect on coronary disease. So I think we're going to see, in fact, I know that there are programs to inhibit both ANGPTL3 and ANGPTL4, and there are programs to try to stimulate ApoA5, all converging on this lipoprotein lipase pathway for reducing triglycerides. Alan, if I have a few more minutes, I'm going to touch on HDL. Is that okay? Or should I stop? I can stop for questions because I think you have an award session, right? Right. Yeah. So I'm just gonna stop here. Anyone who wants to ask can ask me about my opinion on HDL. It's what I talked about yesterday. And the other thing I just want to say while this slide is up is I've talked about lipids, but the idea that we can identify genes that are directly related to coronary disease, not going through lipid pathways, but are directly related to something going on in the vessel wall, is really attractive, right? Because that might identify new pathways that allow us to intervene directly in the atherosclerotic process that, again, would be orthogonal to treating lipids like LDL or triglyceride-rich lipoproteins. So what I've tried to give you a sense for is how the human genetics really can be used to identify new pathways that we didn't previously exist, that are really interesting biologically for scientists who want to try to better understand the physiology and biology, but also many of these are, in fact, therapeutic targets for intervention. So I'll stop there. I'd be happy to take some questions, and then you'll have your award session. So thank you very much.
Thanks. And we actually don't have our normal microphone, so if you just raise your hand, Dr. Rader will call on you.
Alan?
So Dan, as you know, the ApoC3 antisense recently was shown to lower triglycerides in patients and individuals who had no lipoprotein lipase gene.
Yep.
It's an inhibitor of lipoprotein lipase. They have no gene. Could you comment on the mechanism of that?
Yeah, so Alan, I'm going to riff on that question a little bit. So the question basically is, if C3 inhibits LPL, then why did people who are LPL deficient actually have a response to C3? And I think my simple answer to that is that I think it's most likely that those patients who are LPL deficient still have a little bit of LPL, as is true for a lot of homozygous deficient patients. They have mutations that are missensed that still have a little bit of activity. And in fact, if they have a little bit of LPL, they might really be sensitive to inhibition of C3 because it would unmask that LPL and allow it to do more. So that's my leading hypothesis. I'm going to segue from that to say that a similar dynamic exists for homozygous FH and PCSK9. So actually, studies have been done. One study was done with the Amgen PCSK9 antibody in homozygous FH. You should say shouldn't respond, right? Well, actually, the patients in that study who had no receptor didn't respond, as you'd predict. The patients who had a little bit of receptor responded a little bit, but not as well as people who have normal amounts of receptor. So I think, again, the response to these types of therapies actually depends in the homozygous patients on how much residual activity they have of that particular protein. Yeah, hi.
Hi, great talk. You talked about the vessel wall as a target for therapeutic development.
Yeah.
What about the liver or hepatic steatosis? Are you interested in that as well?
Absolutely. So the question is about hepatic steatosis, and of course I touched on that a couple times. And a major unmet medical need, of course. A tremendous interest in mechanisms of hepatic steatosis, but also in therapeutic intervention. And I'm firmly convinced that this approach that I just outlined will point us to potential new targets for intervention for steatosis and NASH and the downstream consequences of that. In fact, that's already beginning to happen. I mentioned TRIB1, where we think that might be a role, and there are several other genes that have been pointed to that are potential therapeutic targets. As I'm sure you and many of us, the path toward registration of a drug that reduces hepatic fat and reduces NASH is very challenging, and the FDA is trying to get its hands around that. But I think the targets are going to come from the human genetics.
So you made a convincing argument for triglycerides in portal in atherosclerosis. Could you comment on the sort of very questionable lack of success of triglyceride lowering and reducing
Yeah. First, I want to emphasize, I don't think it's triglycerides that are causal. I think it's the triglyceride-rich lipoprotein, so the ApoB-containing lipoprotein that contains triglycerides. I know you know that, but just for sake of it. So in brief, you know, the history of triglyceride-lowering agents and their ability to reduce cardiovascular risk is very checkered. Essentially, we have two classes of drugs, fibrates, PPAR alpha agonists, and fish oils. And both have been subjected to trials. The fibrate trials are mixed, frankly. A few have been positive, some have been disappointing. And the fish oil trials are also mixed. But unbelievably, neither of those agents has ever been studied in a large cardiovascular trial in patients identified with high triglycerides, the population that you would expect would most benefit from a triglyceride-lowering drug. It would almost be like starting out with studying statins in people who have normal LDLs. Of course, the statin experiences, the people with the highest LDLs were the ones who were studied first and showed very substantial benefit. So I think basically that's the major issue. I think fibrates are very complicated drugs. PPAR alpha regulates hundreds, if not thousands, of genes. Might have other effects that kind of offset the potential beneficial effects. Fish oils are also fairly complicated. I personally am a lot more excited about the new generation of triglyceride-lowering agents, which I think are much more specific and much more likely to be beneficial in terms of cardiovascular risk. All right, thank you.